Now let's take a look at the shunt step. The shunt step is an important intermediary step that joins the glycolysis process with the Krebs cycle. Because glycolysis occurs in the cytosol and the Krebs cycle occurs in the matrix of the mitochondria, we need to be able to move the pyruvate molecule into the proper location for the Krebs cycle. This is where the shunt step comes into place. Pyruvate is going to be transported into the matrix of the mitochondria. However, pyruvate cannot get into the mitochondria as is. It's going to have to be changed, meaning that it's going to have to lose a carbon to form a 2-carbon molecule. So pyruvate is going to be brought to the surface of the mitochondria, and we can see here that in this diagram we've got the cytosol, you then have the outer membrane, intermembrane space, and the inner membrane. The light yellow area represents the matrix of the mitochondria. So we have pyruvate being shuttled in to the matrix of the mitochondria. In this process, it is going to lose one of the carbons, and that will be used to form carbon dioxide. We also have the involvement of coenzyme A. This is an important molecule that will help to pick up those 2 carbons in that original pyruvate molecule and bring them into the matrix of the mitochondria. In this process, coenzyme A will bind to those 2 carbons and carry them into the internal area. As it does so, it will form acetyl-CoA. The acetyl refers to the 2 carbons, the acetyl group, and CoA refers to coenzyme A. In this process, we also have the ability to generate electron carriers as we form carbon dioxide, and as we're losing that carbon. We can take electrons from that carbon and deliver them to the NAD plus electron carrier. So we have formed NADH in this process, and we have also formed carbon dioxide and acetyl-CoA. Remember that from the process of glycolysis, we would have 2 pyruvate molecules. So this process of the shunt step would occur twice for every glucose molecule coming into the cell. We now have the citric acid cycle. This is where those 2 carbons from the acetyl-CoA molecule that is coming into the matrix are going to be delivered to an important 4-carbon molecule called oxaloacetate. So oxaloacetate is an important molecule to remember. Not the base structure of it, but the characteristics of that structure, specifically that it is a 4-carbon molecule. It is the starting point of the citric acid cycle, and it is the molecule that picks up those 2 carbons from the acetyl-CoA. Because it is a 4-carbon molecule, when it picks up those additional 2 carbons from acetyl-CoA, it will now become a 6-carbon molecule. This 6-carbon molecule is citric acid, which is why the citric acid cycle is named as it is. As the cycle turns, because this is a cycle, these carbons that are coming in are going to be completely oxidized to form carbon dioxide. And this carbon dioxide is eventually going to be released by the cell. So there's 2 important things that happen. One is that these carbons coming in are going to be oxidized, and 2, that we need to regenerate this 4-carbon molecule. Because this is a cycle, we have to make sure that we are regenerating oxaloacetate. And that way, that oxaloacetate can then continue to pick up another acetyl group, so on and so forth. The citric acid cycle, in general, makes a lot of electron carriers. 1 turn of the cycle, meaning that 1 acetyl-CoA group coming in will produce 3 NADHs, 1 FADH2, 1 ATP, and 2 carbon dioxide molecules. Remember that glucose is broken down into 2 pyruvate molecules, so that means that we would get 2 turns of this cycle. So we would generate 6 NADH, 2 FADH2, 2 ATP, and 4 CO2 molecules for each original glucose. This image here depicts the steps of the citric acid cycle. Once again, I do not expect you to memorize all of the structures and all of the enzymes and steps involved in this process. I will take you through what is important to know. What I would like you to do is trace the carbons throughout this process. Understand what is happening when we are generating carbon dioxide, and understand that overall the citric acid cycle is made to produce many electron carriers. Eventually, these electron carriers are going to be delivered to the electron transport chain to indirectly make ATP. So the citric acid cycle is a very important step in making all of those electron carriers and harvesting all of those electrons from the sugar molecule. So here we've got acetyl-CoA coming in to the cycle. Note that it has the 2 carbons that originally would have come from the glucose molecule. We have oxaloacetate, a 4-carbon structure that is coming in to pick up this acetyl group. So coenzyme A will leave, and these 2 carbons will then be attached to oxaloacetate to form a 6-carbon molecule. This 6-carbon molecule is called citrate or citric acid, which is why the cycle is named the citric acid cycle. This will then undergo a series of isomerization reactions to form isocitrate. So all that means is that the carbons are simply going to be switched around so that enzymes can effectively work on it to cut off those carbons, cleave those carbons, extract those electrons from it to eventually oxidize all of those carbons in the pyruvate or glucose molecule. So this is where that electron extraction occurs. When we're going from isocitrate to alpha-ketoglutarate, this is what I want you to remember. We are going from a 6-carbon molecule to a 5-carbon molecule. That means we are losing a carbon. If we're losing a carbon, that means that we are forming carbon dioxide as a waste product. This also means that we have the opportunity to take electrons away from that structure. So that means that as we make carbon dioxide, we can also generate an electron carrier, and that is what we see here. As we're switching from a 6-carbon to a 5-carbon, isocitrate to alpha-ketoglutarate, we are making carbon dioxide, and we're also generating an electron carrier. This occurs again. We are going to go from a 5-carbon molecule to a 4-carbon molecule, alpha-ketoglutarate to succinyl-CoA. Count the carbons, 5 to 4. And as we do that, once again, we can produce carbon dioxide, we can take those high energy electrons, and we can generate an electron carrier. So this is typically what we see when we're going from a 5-carbon to a 4-carbon, when we're going to 6-carbon to 5-carbon, we're going to make carbon dioxide and generate electron carriers. This brings us now to a 4-carbon structure. This structure is called succinyl-CoA. Now we have the regeneration phase of the citric acid cycle. This means that we will have to undergo a series of isomerization reactions to regenerate our important oxaloacetate. So notice, all we're doing is rearranging those 4 carbons. So in the second half of this cycle, we do not see carbon dioxide produced at all. What we do see, however, is our electron carriers being generated. You can also see that we are generating a little bit of ATP in that first isomerization step. So yes, we generate a little bit of ATP in the citric acid cycle. It's made indirectly. We actually generate GTP first, and then that is transferred to ATP. But overall, the goal of the citric acid cycle is to make all of these electron carriers. So we can see through the isomerization of that 4-carbon molecule to regenerate oxaloacetate, we are producing FADH2, and we are also producing NADH. So more electron carriers. Keep in mind that we have now regenerated oxaloacetate, so this cycle can continue. Also keep in mind that this cycle, represented here on this diagram, is illustrated for one acetyl-CoA molecule. If we started with a glucose molecule, we would have 2 pyruvates generated, which means we would have 2 acetyl-CoAs generated, which means that this cycle would occur twice. Now we have our last step, which is oxidative phosphorylation. So keep in mind, through all of the steps we've seen so far, glycolysis, shunt step, the Krebs cycle, only a little bit of ATP has been produced. But through the Krebs cycle, we have generated a lot of electron carriers. NADH primarily, however, we've also generated FADH2, simply another electron carrier. These electron carriers are carrying high energy electrons. And remember, when they are passed to NAD plus and FAD plus, we have them passed in the form of that H minus or hydride ion. But all of these electrons are high energy electrons. The overall goal of this oxidative phosphorylation step is to harvest that energy from that electrons and make ATP molecules. So this is where the bulk of ATP is actually going to be produced. This occurs once again in the matrix of the mitochondria, but all of the enzymes present are going to be present in the inner cristae, or innermost membrane of the mitochondria. How this occurs is by utilizing those electron carriers, NADH and FADH2. They're going to pass off their high energy electrons to the electron transport chain. That energy in those electrons is going to be used to pump protons. And this is occurring, remember, in the innermost membrane of the mitochondria. So it is important to review the overall structure of the mitochondria because it's important to understand where these electron transport chain and where these electron carriers are becoming important, and also where these protons are being pumped with respect to what space. The electron transport chain is then going to allow for these high energy electrons to pass through it. As these high energy electrons pass through the electron transport chain, their energy is going to be used to pump protons out of the matrix and into the intermembrane space. This is going to create a proton gradient across the inner membrane. This is the important first step of oxidative phosphorylation. So it is important to note that we're not directly producing ATP. There's one important thing that has to happen before ATP is even produced. We have to have a proton gradient. Without that proton gradient, we cannot produce any energy. Once we have that proton gradient established, remember, in establishing a proton gradient, this is considered to be active transport. So we are going to use energy to pump these protons because they need to go from a low area of concentration to an area of high concentration. This energy will come in the form of the electrons. This is very different from what you've seen before, and a lot of students get trapped in this concept. So the energy is going to be carried in electrons. This is going to supply the energy to drive active transport, to drive those proton pumps in the electron transport chain, to then create that proton gradient. Once we have that gradient established, those protons are going to flow back through the inner membrane. So they're going to flow from the intermembrane space back into the matrix. They're going to do this through passive transport because they're going from an area of high concentration to low concentration. This is called chemiosmosis. This is where ATP synthase, an important enzyme that is involved in ATP synthesis, is going to take advantage of that proton gradient. As those protons flow back through passively, it is going to harness that energy and use it to form ATP molecules. This ATP synthase works much like a turbine. So as the protons flow, the bottom part of the enzyme will be able to move and turn like a crank. And eventually, once you get 3 of the protons coming through, there's enough energy in that turn to then be used to form an ATP molecule. If we go back to the electron transport chain for a second, it's going to be going from a high energy state in those electrons. It's going to use some of that energy from the electrons. So as the electrons pass through, they're going to be losing energy as they pass through the electron transport chain. As a result, when they get to the end of the electron transport chain, they're going to have very low energy associated with those electrons. And those electrons are going to need a final destination. In this instance, this is where we have oxygen coming in. It is going to be the final resting place for those low energy electrons. So those low energy electrons are going to be passed to oxygen in the form of the hydride ions, and this will then lead to the formation of water. So this is considered to be an aerobic step because oxygen is being used. In this case, however, it's being used to take in those low energy electrons from the electron transport chain. This diagram here illustrates the different protein complexes in the electron transport chain. Notice that we have 4 different complexes in the electron transport chain. Complex 1 is responsible for taking in those electrons from NADH. So NADH is going to come to complex 1, give its electrons up, become oxidized to return to NAD+. Those electrons are going to then pass through the rest of the electron transport chain. So the yellow arrow here illustrates the passage of electrons. These red diagrams here illustrate the protons and where they're being pumped. And the black arrows here represent what is being formed. So here we've got NADH dropping the electrons off and forming NAD+. Those electrons are going to pass through complex 1. As they pass through complex 1, they're going to use some of their energy to pump protons. So this is why we see some of the protons being pumped. Remember, the orange area represents the intermembrane space. These phospholipids and proteins represent the inner cristae or inner membrane of the mitochondria. And this yellow area represents the matrix. Now draw your attention to complex number 2. Complex number 2 of the electron transport chain will take in electrons from FADH2. So FADH2 will drop off those high energy electrons and form FAD. These electrons from complex 1 and complex 2 are then going to go to coenzyme Q. So this is simply an accessory coenzyme that will help transfer electrons. So whenever you have an electron transport chain, it simply allows electrons to flow through, and in this case, a series of proteins. So we have electrons coming in from complex 1, electrons coming in from complex 2. However, notice that in complex 2, there's no protons being pumped. So complex 2 can only take in electrons from FADH2. It doesn't pump protons at this point. Those electrons from complex 1 and 2 are going to be passed from coenzyme Q onto complex 3. Notice that complex 3 is not taking in any new electrons. It's simply taking those electrons from complex 1 and 2 and allowing them to pass through. Once again, the energy from those electrons are going to be used to drive the proton pump. So protons are going to be pumped from the matrix into the intermembrane space. Remember, this is active transport, so you should be keeping in mind that there's a low concentration of protons in the matrix, and we are building a high concentration of protons in the intermembrane space. These electrons are then going to be passed to cytochrome C. Cytochrome C will simply allow those electrons to then be passed onto complex 4. In complex 4 of the electron transport chain, we have a last opportunity to extract the last remaining amount of energy from these electrons to pump protons. So once again, we will see protons being pumped from the matrix to the intermembrane space. In complex 4, we also need to have a final resting place for those low energy electrons. At this point, oxygen is going to come in. Those electrons are going to be passed to hydrogen to form the hydride ion. This will then bind to form H2O. At this point, all of the energy from those electrons that originally came from the glucose molecule are now going to be utilized to create that proton gradient. So at this point, they're considered to be very low energy when they do form water. Now let's take a look at what happens next. So this is the first part of the oxidative phosphorylation, where we are using the energy from electrons to create a proton gradient. Now that this is established, now we can consider making ATP molecules. This step here illustrates ATP synthesis. So once again, in order for this to occur, we have to have that proton gradient established. Once that gradient is established, we can have chemiosmosis occurring, where those protons are going to flow back through from high to low concentration. So they are going to go from the intermembrane space, the high area of concentration, to the matrix, where it is a low concentration of protons. This is going to be done passively through the enzyme ATP synthase. As these protons flow through, ATP synthase is going to turn around a bit like a turbine, and once you get enough protons flowing through, that is going to lead to the production of ATP. So ATP synthase will take ADP and pi from the matrix and join it and take that energy from the protons flowing through and harvest it into the ATP molecules. Remember, we're making a high energy bond in that ATP molecule. This provides a summary table of what is being made in these processes and where these are being produced. So glycolysis, remember, occurs in the cytosol. Here we are taking one glucose molecule, we're using some ATP to tag it so that it can be broken down properly, and we're also using some NAD+ because we need some electron carriers to carry some of these high energy electrons. This is going to be used to produce 2 pyruvate molecules. We have 4 ATP molecules being produced and 2 NADH electron carriers. The shunt step uses those 2 pyruvate molecules. Once again, we're going to be using NAD+ because we need to generate some electron carriers, and coenzyme A is going to be required to carry those 2 carbon molecules into the matrix of the mitochondria. So the shunt step, in that step, we're going to be producing 2 acetyl-CoA molecules. We're also going to be producing 2 carbon dioxide molecules because we are losing a carbon off of each pyruvate. And we're also generating some electron carriers as well. Every time we produce a carbon dioxide, we can generate an electron carrier. And we have 2 pyruvates coming in, so we have the opportunity to do that twice. In the Krebs cycle that occurs in the matrix of the mitochondria, we're taking 2 acetyl-CoA molecules. We're also going to be taking in a lot of oxidized electron carriers because we want to eventually reduce them so that they're carrying those high energy electrons. So we're going to use 6 NAD+, 2 FADH+. We're going to be generating a lot of carbon dioxide, 2 molecules of carbon dioxide for each turn of the cycle. And once again, we have 2 acetyl-CoAs coming in, so there's going to be 2 turns of that cycle. So we're going to produce 4 carbon dioxides overall, 6 NADH, 2 FADH2, and also a little bit of ATP indirectly. Remember, when that 4 carbon molecule first isomerizes, we are making GTP, which then will make ATP. And finally, we have oxidative phosphorylation, where we're going to be taking all of those electron carriers, 10 NADH, 2 FADH2. We're also going to be using some oxygen. We're going to be making water molecules from that are carrying those low energy electrons, and we're going to be generating a lot of ATP in this step. So here we have an overall depiction of how these reactions correlate with each other. So we have glycolysis occurring in the cytosol. We're going to take that glucose molecule and we're going to convert it into 2-pyruvate, making a little bit of ATP along the way and generating a few electron carriers, 2 NADH molecules. These NADH molecules are eventually going to go to the electron transport chain. We then have pyruvate oxidation. This is also called the shunt step. So we're oxidizing pyruvate in the shunt step to produce acetyl-CoA. So with pyruvate oxidation, we're taking pyruvate, we're converting it to acetyl-CoA, and that acetyl-CoA will then enter into the citric acid cycle. In the citric acid cycle, we are going to be generating the bulk of those electron carriers that will then drop off their high energy electrons to the electron transport chain. Once again, we create a little bit of ATP in that process when you first start isomerizing the 4 carbons to regenerate oxaloacetate. And this occurs in the matrix of the mitochondria. So the pyruvate oxidation, or the shunt step, joins the products from the cytosol and brings them into the matrix so the citric acid cycle can occur. Oxidative phosphorylation will occur using the electron transport chain, generating that proton gradient, allowing for chemiosmosis to occur for ATP synthase to generate the ATP molecules and harvest that energy.